Hello, friends. Today we're going to try to get you ready for the Ohio Biology State Test. We're going to do that by looking at past questions from the Ohio Biology Test. The questions we're going to look at today are from the year 2016. So let's begin. DNA samples taken from rats from the same colony produced the 4 gel electrophoresis banding patterns shown. Determine the relationship between rat A and the other 3 rats tested, B, C, and D. Place relationship labels into blank boxes under the electrophoresis pattern for rats B, C, and D. Place only one label in each blank box. You may use each label more than once. So we have a question here about electrophoresis. Remember what electrophoresis is. It's a way we can identify DNA. For example, DNA molecules separate in a certain pattern. So if you have similar DNA, you have similar patterns. So remember, electrophoresis, we have a gel. We put the DNA samples at the top, and we put electricity through it, and the molecules of the DNA separate out. And we can compare them, and that's what we're doing here. So we're going to compare rat A to rat B, C, and D. So what is the relationship to rat A? Well, we got to find something that's very close to rat A. If we look here, B doesn't look very close. C does look pretty close. And D looks, but C is very close to rat A. So we're going to say that's an identical sibling. Now, let's see if we can find a sibling or a parent. Now, a sibling or a parent's not going to have the exact copy of the DNA, but it's going to be close. It's going to be similar. So rat D also has a thick line at the top. It also has a band here too, okay? So I would say rat D is like a sibling or a parent to rat A. And if you look at rat B, there's hardly any similarities. So it would be unrelated. And that's how you do the first problem. Now, let's continue with our next question. The table shows a number of amino acid differences among organisms A through E for a common protein with 113 amino acids. Use the data in the table to complete the cladogram by placing an organism label in each box. Place only one organism label in each blank box. There may be more than one correct answer. So if you look over here, we have A, B, C, D, and E, okay? And if we look across here, this is the differences in amino acids. Now let's remember what an amino acid is. Remember when we talked about from DNA to RNA to protein, DNA had the instructions for RNA to make amino acids. And what are amino acids? They're the building blocks for protein. So the closer your DNA is, the closer your amino acids are going to be. So we look here, and of course we're comparing the differences. And of course there's no difference between A and A. Now there's 11 differences between B and A. There's another 11 differences between B and A, C and A. There's like 28 differences between D and A, and you can see there's a lot of differences between E and A. So let's look at our cladogram. Normally, the cladogram, this box would be over here, and you could see it. Remember, a common ancestor, when you have a common ancestor, that's when your DNA is closer together. So let's look here. We have B and C very close. We have D here, which is going to pair with something else. Well, since D is down here with one common ancestor, a common ancestor here would be for B and C. They would be closely related with their DNA. Now, what would be closely related to D? Well, that's the only one left. That's E, okay? Now, since we begin with A, that's the one that we have left over. And so A would come after here, okay? Common ancestor, B and C close together. That's true. There are only 11 differences from A. And E and D are very close, and A is where it's all started. Okay, here is our next question. DNA sequence for a number of bird families in the order of Passeriformes are shown. Difference in DNA sequence between the Ptiloris family and other bird families in this order are highlighted in red. An incomplete phylogenetic tree that notes important differences in DNA among these bird families is also shown. Use the DNA sequence data to complete the phylogenetic tree. Move each bird family labeled into the correct blank box in the phylogenetic tree. Now, there's a lot of big words here. I don't want you to worry about them, okay? You can use just their letters, okay? You don't really have to be afraid of these names, okay? So let's look what we're after. This is kind of similar to what we just did. Remember, DNA, the closer you are in DNA in the order of your bases, the closer you are related, okay? The closer your DNA is, the closer you are to being related. So we have, again, a phylogenetic tree, and here's our Ptiloris, and you can see it's all black. Where there's a red letter, that's a change, okay? So, again, common ancestor here in our phylogenetic tree. Let's just see what happens. So we can see here the changes. In 26, so we go over here, 26, and T becomes C. Whoa, you can see here 1, 2, 3, 4. That doesn't really help us. So let's go over here. In 13, C becomes A. There's 11, 12, 13, and C becomes A. Look, there it is. So it has the A, and there's a C. Well, there's our word. We're going to just put that little birdie here, okay? Because that's what avian means. Avian means bird. So now let's look at our phylogenetic tree again. So we have the 26T becoming C. We see that. We have the 1 becoming a C, okay? So we're over here. And then we have a 31C becoming T. So let's find 31. And look, there it is. It's the bird that begins with D. And we'll put that in there. Okay. So now let's go back here and see what else. We got the 1T becoming C. So we're over here. And then, so what are we left with? The number 1T becoming a C. Which one didn't we use? Well, the one's left over. And see, seemed hard. A lot of big names. Had to move things around, but it really wasn't that hard. Here's our next problem, and it looks like the electrophoresis is back. Scientists have found an allele of a plant gene that may protect plants from some types of insects. To determine whether this allele is present in other plants of the species or is a recent mutation, they conducted the following experiments. Okay. They isolated a sample of deoxyribonucleic acid, we know that as DNA, known to contain the allele from one plant that is unaffected by insects. Isolate a sample of DNA that may contain the allele from 3 plants of the same species grown in the same location. Treat the resistant DNA sample and the 3 DNA samples from other plants with the same restrictions enzymes to generate smaller segments. Load and run a gel electrophoresis to separate the DNA segments. Analyze the results of the gel electrophoresis. Okay, so we got to figure out which one of our samples, okay, is similar to the DNA resistance sample. So if you look here, this is what we're comparing it to. Sort of like that first problem we did. Sample 2 sure looks like it. There's our answer. Sample 3 doesn't. Okay? Again, looks hard. A lot of reading, but not hard. Here's a problem labeled number 5 from the 2016 state test. How could genetic engineering be used to produce a more successful crop in a hot, dry climate? Ooh, genetic engineering, we'll be fooling around with some DNA. Well, here's our choices. Non-essential DNA could be removed from the genomes of the crop plant. B, DNA from plants adapted to dry areas could be added to the genomes of the crop plant. C. RNA from a variety of non-agriculture plants could be added to genomes of the crop plants, and messenger RNA transcript of genes from dry weather crops could be added to the fertilizer used on the crop plants. So I don't understand why non-essential DNA would help a plant exist in a hot, dry climate. In other words, make a plant more acceptable to living in a hot climate. I don't see that happening. RNA from a variety of non-agricultural plants could be added to the genomes of crop plants. Messenger RNA from genes from dry weather crops could be added to the fertilizer. Guys, here's what's happening. The true answer is B. DNA from plants adapt to dry area could be added to the genome. So we're taking the DNA, parts of the DNA, from one plant that lives in a hot climate and putting it into a plant that isn't able to exist in a hot climate. Okay? So that is how genetic engineering works. We're taking DNA from one plant and putting it into the other. Okay, this is the kind of problem I like, okay? Because it has to do with DNA to RNA to protein. A mutation occurs in the DNA base sequence GGG, GAG, TTA, resulting in the base sequence of GGA, GAG, TTA. Use the information in the table to determine the effect of this mutation on the amino acid chain produced. Okay, so we have, this is our DNA base sequence. And we know it's DNA because they tell us it's DNA, but it also has a T in it. Remember, RNA doesn't have a T. So what we need to do is convert our DNA into RNA and then see what amino acid corresponds with that RNA. So if we look here, we got GGG in our first DNA. The mutation has changed it from GGG to GGA. So let's look up the amino acid for a little. First of all, we've got to turn our DNA into RNA. So GGG would be garage, cars, garage, cars, garage, cars. We'd have 3 C's. Look up 3 C's. There we are. Proline. Now, let's look where the mutation has occurred. We got GGA. So that would be CC. Remember, there's no T in RNA, so that would be U. So CCU. Oh, even though the mutation has occurred, it still hasn't changed our amino acid. Okay? So now, let's see if these are all the same here, GAG and TTA. So let's see, GAG would be CUC. CUC is leucine. That would be the same over here. And then TTA would be AAU, asparagine. Okay, so let's see what our questions are. What is the effect, if any, of the mutation on the chain produced? Well, we know our proline is going to be unchanged, so that's that one. Okay, now we know our proline's unchanged. We also know AAU, that would be this, is also, the asparagine is also unchanged. So there we go, okay? Okay, because the leucine didn't turn into phenylalanine, okay? And we know the proline didn't change, so there is our answer. Again, you think it's kind of hard, but it isn't when you sit there and think about it.